All right, thank you everyone for coming to the Friday Ecology Seminar. It is my pleasure to introduce and our privilege to have Professor Nico Franz and give a talk today. He is the new director of the Biodiversity Institute. Came here from Arizona State University. His taxonomic training is in entomology, and he has a great fondness for weevils and already been out to the field station to begin researching weevils. So today he's going to talk to us about Symbiota, which is amazing infrastructure that he has brought to KU. So with that, Nico.
Yeah. I'll share my screen in a second. Thank you, Sarah. Thank you, KBS community. Yeah, it's my privilege to be here. I will just say that this talk will be a little stitched together, maybe frantic, but I'm gonna try to pay attention to what may work for you and what you need. And so it is supposed to be somewhat interactive. I don't have a slide set for you today. I think a high level outcome is to plant enough of a seed that if you think that there could be follow-ups, then we can have those follow-ups that might advance your research interests and needs and so forth. Just a little bit about myself. Yes, I'm trained as an insect systematist, but in my previous sort of career at Arizona State University and continuing here, one research specialization that I have, if you can call it that, is to sort of design, implement, and advance research infrastructure that can include physical infrastructure, think biorepositories, and sort of flows in, flows out, services around that, and also virtual, aka data infrastructure. And a general threat in building infrastructure is to pay attention to social practices or social relationships, power dynamics, if you will, access, rights, governance, integrations that we hardwire in one way or the other when we build infrastructure, right? Infrastructures are an expression of social contracts and social dynamics. And when one has the opportunity to design and to implement infrastructure, paying attention to that, I consider that part of my research program. And let me also say that I have really appreciated, valued, and benefited so far, and it's only going to continue and grow from my interactions with Sara. I think we've been very good colleagues, and we have plans separately and together to keep growing. I'm very impressed and fond of the KBS community. I think from what I can see so far, your research programs are amazing. Your sort of research output and productivity is really stellar and impressive and commendable. I also appreciate the sort of service to our community, both within KU and within the state and beyond, that KBS provides. So in that sense, I have some background in ecology, but even if I didn't, I just think that you are an awesome community, and in that sense, I'm grateful and fortunate to be here. Okay, now I can share my screen. All of it. And so talking about Symbiota, which is a biodiversity data management and publication platform, could start with some conceptual issues, and then we talk a little bit about how it works, or we could just dive right into it. And I decided for this talk to just kind of dive right into it in a somewhat idiosyncratic way. But I'll just start with like a 3.5-minute video about Symbiota that covers a lot of ground very quickly. Okay, so that was a quick start. And in the wake of today, we just put together a 2-page flyer that gives an overview, sort of at a glance. What is this software? A little bit of sort of usage stats. So by now, close to 2,000 natural history collections, broadly defined, live in our 60+ hosted portals, we're nearing 100 million vouchered sort of records, and a little bit of powering toward the blue, which is KU's new brand. We're repping for the state, you know, to the tune of close to 500K records and so forth. And there's contact information there. We have a active help desk, so a really good way to reach out, in addition to sending me an email, which is totally fine, or finding me on Teams, which is very successful. The Symbiota.org and the Help Symbiota works. And here are just some more updated than this video sort of impact metrics that we have to report in part to the National Science Foundation, where we are a sub-awardee of the IDIC Biodiversity Hub. And so this sort of characterizes you know, the number of active users that we have, the number of occurrence or specimen record edits that we have received, how many tickets we enter, and so forth. And most importantly, it's nice to put a face on the team. So this is the team that in the second half of 2024 fully transitioned from Arizona State University to now be employees with the Biodiversity Institute at KU. So that includes software engineers. Dr. Samantha Oriana is our Latin American outreach coordinator, project management, IT management, community outreach team that we are trying to sustain and move forward. And speaking of sustainability, this is also just a little vignette. If you search for KU Symbiota, by now you would find us right here. I can make this a little larger. And so we have a sort of homepage in the Biodiversity Institute page environment, but more importantly, we are largely grant funded, and we are in the process of diversifying our revenue model to not just sustain but grow. And one of those revenue streams is we are also a KU so-called service center. So we provide fee-based services in addition to working with grants and contracts and so forth. So products that we have include image hosting, data portal setup. So if you wanted a new Symbiota portal, the price is actually available online. If you wanted us to help manage that portal, we can do custom development. We do custom data sets and other type of consulting. And I wanted to mention just how much this is by now part of KU and KU sort of infrastructure and services, also accessible to KBS and the greater community. So then I wanted to kind of jump into an actual experience with Symbiota, and I'm going to try to do this somewhat slowly and not to overwhelm anyone, but you may get vertigo, and I may also really have to pay attention just to how many tabs I have open at a time. I'm trying to pace myself here, right? SciNet is one of the 60+ portals, and by now you understand that the core and the boundaries of these portals are community defined and community owned, right? We don't make the focus or the constellation of any portal. This portal was originally located sort of in the Southwest, and it deals with herbaria. And so it's got a map search function that is generally considered user sort of accessible. You know, if you ask yourself, as I'm sure you do on a regular basis, just how many members of the sunflower family occur in Lawrence, right? And it took me a little while to get there, but now I'm here. You can use a circle and you sort of hover over that. Maybe you have to move it a little bit. That's my Lawrence circle. Does anybody want to guess how many species of Asteraceae, the sunflower family, in Lawrence? Yeah, not correct. Not cool, right? Let's see some of our other survey experts here. Bring it. Yeah. Yeah. Let's find out, right?
Does it include cultivated stuff?
I heard 100, right? You can instantly see that there is, well, you can't, but you can, I will let you know that there's very sophisticated taxonomy management under the hood here in terms of synonymies and multiple representations and so forth, right? This is far beyond anything that you could do, you know, in a spreadsheet format or something like that. It's a complicated and finely interacting sort of data model. But let's do our search, and we get some dots. And so the collaborative aspect of the software, 243 records, right? That's not species, that's specimens, right? Think pressed plants in herbaria, and you can download these variously. But the collaborative aspect, you know, is, for example, exposed by these are the different source collections that have contributed these close to 250 records. And so this is not just KU, right? This is a community that defines itself beyond conventional institutional boundaries, right? So this could be all of the LTR plant collections or something like that, right? Or all of the long-term ecological research communities in the state of Kansas that have agreed to somehow come together in this portal, right? And the answer is 105 in this particular search. And if I wanted to auto-color them, I can do it like that, right? And if I wanted to look at a particular one, now my tab management already needs to come in, because I tend to get pretty jittery with this. So I selected one of the 105 species there, which happens to be the perennial ragweed. And you can sort of see that there are the kinds of photos that we can, for example, import from iNaturalist. We would call them observations. But by the time I get to all images, that was not what I wanted, or is it? Yes. You see that much of this is driven by herbarium specimens, right? That have been databased in terms of their collecting event and taxonomic data. They have been imaged and they have been published through this portal to a broader community. And I'll let go of this one. I'll just click here. So then for each one of these records, Symbiota has, of course, a vignette of associated information. And I hope that at least some of you will be familiar with the term the Darwin Core Standard. This is the data standard that is prevalent in the global natural history collections community. And I know something about data standards in ecology, for example, ecological metadata language and derivatives of that. From my perspective, the level of commitment of the global collections community to this particular standard Darwin Core is a real gift that we have somehow given ourselves, and it has allowed us to be really, really strong in integrating natural history collections data across the globe, and also to further manage that standard and its implementation. But Symbiota has sort of this default design that it's based upon the Darwin Core. That doesn't mean that that's all it does, but it's sort of a minimal common denominator when we engage with new communities. Can at least significant parts of your data be meaningfully represented in the Darwin Core standard? And if that's the case, then we can work with very often whatever system or environment your data in right now, and we can start to work to potentially import it and ingest it and represent it in Symbiota, if that makes sense. Now I'm going to jump very quickly. You can perhaps see that the relative ease of sort of navigating this system, and also the fact that you can build checklists for a particular taxonomic group in a particular region lends itself to a lot of educational opportunities with these collections-based biodiversity data. None of what I've shown you so far would have required any sort of login, and that's kind of critical. But now I want to jump from plant data to the NEON Biorepository data portal. So this is also a Symbiota portal. NEON, as many of you will know, is the National Ecological Observatory Network. It is a NSF major facility, one of 18 and the only one in bio, right? And it's ecological monitoring and forecasting is the center. NEON produces something like 182 data products routinely across the 81 field sites that it operates. And about 70 of those are physical samples that are being vouchered in the NEON Biorepository. And so when we started to work with NEON on the Biorepository at ASU, both physical and data, we expanded and customized Symbiota very significantly to be better at managing ecological collections, right? A couple of ways that I can just quickly illustrate this. So if you go to sample search here in the NEON Biorepository data portal, and you just look at the kinds of collections that we have here. Again, let me make this a little bigger. You recognize that we have now expanded the scope from conventional natural history museum specimens to ecological samples, right? Which has meant that we've had to create sort of parallel, in computer science, you might call them ontologies, hierarchies of classes and relationships in order to be able to say, some of our samples are taxonomically indexed and others are maybe indexed because of ecological categories such as particulate mass filter or benthic microbe collection and so forth. And going one further, now I'm going a little bit under the hood and I'm just going to pick a specimen. And this is a big theme with Symbiota, is we're building a lot of extensions from the conventional natural history specimen to related instances or data packages related to that specimen, right? What I'm showing you here right now is sort of the front page of some kind of, what are they called? White-footed mouse, Peromyscus, right? So we have images here, right? I'm not going to pop this up because I'm getting into tab issues, but you can blow this image up and you have a very, very large image of that specimen. This links to the main NEON portal maintained by the NEON operator Battelle. There are material samples related to that specimen. So there's a skull preparation. There's a postcranial skeleton preparation. There's credit here as to who made this preparation. There are various notes related to this particular specimen. It may have gotten barcoded with CO1 DNA. Lots of traits. Some of these are free text, but following a certain format, but there are also capabilities to really have structured language representation when you get to traits. Detailed locality information, taxonomic identification, history attributed to various folks. In the natural history collections community, this is called the extended specimen network or the extended specimen paradigm. And just in terms of biodiversity data and sample infrastructure development trends, that's where that community is moving to move more from the idea that one repository has it all to we need to invest into these repository to repository linkages and integrations, right? So other integrations include that this particular mouse was the host of MEEP with reference to the Roadrunner, mammal endo and ectoparasite collections, right? So I don't think we have an image of this particular water siphonaptor. Flea, yeah, right. But flea has host versus mouse is host off. So entity, relationship, entity is the basic building block of this specimen concept, extended specimen concept. You have your geographic data, you have a link to GenBank or BOLD in this case for DNA barcoding. You have other linked resources, for example, whether it occurs in a particular inventory or not, whether it's a member of other data sets. You can annotate traits. This is pretty rudimentary here, but there's more capability. And now I'm jumping from everything that you can see on the front end seamlessly to the back end. Symbiota is never installed on anyone's desktop, right? It doesn't do that. So what you see here would be the contributor or editor interface that the researchers managing this particular sample are having. And people who work with Symbiota generally like the fact that everything that you have to record with regards to one sample is just one page, which you see right here. You can see other capabilities here, right? So for example, the ability to manage multiple identifiers. All of your ecological samples that you sort of care about in the longer term will have some sort of identifier, right? That can be carried over, but then there can be globally unique identifiers, so-called IGSN, international, what is it? Generic sample number can be associated with these. There's a lot of technology built into the identifier management. And then this is straight up Darwin Core with the different fields and their relative. These question marks actually go to the Darwin Core definitions and so forth. But you can see that there's tabs for determination history, there's tabs for image management, there's tabs for adding and editing the material sample. There's other linked resources. So here you have your associated occurrences, right? And if I go further down here, actually, let me quickly see. If I wanted to create another sort of resource, I can import different kinds of ontologies. These happen to be neon customized, but they could be very much customized for whatever project you have because the structure of the software is present. Okay, and for the fun of it, I also wanted to mention that these Symbiota data sets can be put out to the Environmental Data Initiative. Some of you may be familiar with that, right? EEI, they publish. My understanding is that sort of the development of the LTER community in our country at some point motivated this idea we need a relatively centralized, open place to publish data so that they could be adequately retrieved and published, right? And so most of the Symbiota collections that are managed in the NEON portal are compatible with this aggregator called GBIF, but that is conventionally a natural histories collections type aggregator, right? And then there's some 30 collections that now work in Symbiota. They're very ecologically circumscribed. They no longer work for GBIF, but they still work for TEI, for EDI. And the benefit there being that if the NEON Biorepository has a total of about 70 collections, 40 of them fit into GBIF, they all have a DOI there, digital object identifier. The others fit into EDI, they have a DOI there. So it's at this point increasingly hard to ever work with NEON Biorepository data that's managed through the Symbiota portal and not get a DOI based on which we then can show our impact metrics, right? And that's an important part of the design feature. Let me close a few things I'm not going to go on and on. I feel like I've covered enough ground for us to have a Q&A pretty soon. But I wanted to talk a little bit about attribution. And this is an insect portal. And this is the collection homepage for an insect collection that was generated by an undergraduate student who was at the time trained with us at ASU, right? So you don't have to be the Biodiversity Institute or the Smithsonian. You don't have to be a faculty or a staff. You don't have to be a postdoc or a graduate student. You can be an undergraduate student who likes to collect insects, and you can get into a portal, right? And Evan, who's now a graduate student at University of Central Florida, happens to really like collecting ground beetles and other specimens. And so we were able to open up for Evan a collection in this particular portal. And that then means that Evan's collection does have a DOI. And it does, for example, mean that it has by now amassed 72 citations because, as I will show you, and these are them. So this is an undergraduate student collection, now graduate student, that has created enough data that people who have accessed this data through GBIF have basically cited parts of it 72 times. And this gets back to the notion that here's an integration of that same collection with the global aggregator that I think by now surpasses 3 billion of these kinds of Darwin Core compatible records and basically published here. And so what that means is that it's a very low barrier of entry by design, and it lends itself to participation and contributions by many communities that would otherwise not be able to participate and also, you know, sort of get credit for their data and for their contributions in these sort of global frameworks. I have one more that I'm sort of hesitant about, but I'm just kind of going to try to do it, which is a new, there are a couple of developments that we are focusing on sort of topically now and in the near future. One of them, and again, I'm probably not connecting because we do have sort of our own language. Where is it here? Is known in the collections community as the Humboldt extension, right? That's an extension of the Darwin Core that is being worked on here by a task force. And in a nutshell, it builds out how much we can say about collecting events, irrespective of whether a sample was taken or not, right? And if I had to make that much more simply, absence data, right? Natural history collections historically have not had good infrastructure. It has not been a good focus to record absence data. But this is clearly a motivation and also a signal to connect better with ecological monitoring, because absence data is a critical component of many ecological sort of monitoring and forecasting sciences, right? And so if you record more data and also are able to expose data better about the collecting event, then you can basically say, we went there and we found something, then we went there again and we didn't find it, then we found it again, and so forth, right? And so this is something Symbiota, through the vehicle of NEON, is trying to push even more into providing services for long-term ecological research projects, and being one of the strongest software sort of out of the gate to really represent absence data is our strategy to be able to make that connection better. The other one is data ethics, right? Or data governance or data rights. And one of the things that you can do when you have a very rich community of natural history collections, all contributing data under fair principles, open everything, access everything, all of a sudden you can put together, and this list is protected, it's not publicly visible, you can only see it because I'm logged in and I'm a super administrator. That in itself is not trivial or necessarily okay, but we can technically create a shapefile that somehow, give me a second here, aligns with boundaries, for example, of a native nation, in this case, the Tono O'odham, in central South Arizona. And we can represent all of the data that is there potentially. While this is coming, I'll show you just one other aspect, because I also know we have some ethnobotanists in the room, right? These are things that just for my own biography, I sort of slowly discovered. One of the herbaria that's in the SciNet portal, and it's kind of stalling right now, which is regrettable, is the Navajo Nation herbarium. And so that means we have, I'm sorry about this. We actually on this particular server have experienced a high load just today. And so I just kind of went for it. The point being is that you may also have heard of the CARE principles. I'm sure you've heard of the FAIR principles, right? The CARE principles at a high level deal more with ethics, collective benefit, governance, and restraining access, and also potentially propagating these kinds of restrictions from one form to the other. And this is something that we do want to work in, because even though the maps here are stalling, you know, we basically can represent for an entire native nation how many plants have been recorded, none of which are currently in herbaria that you native nation governments have any control over. We have some herbarium specimens that clearly show cultural knowledges and related information. And just last week, was it just last week? KU held a 2.5-long summit on indigenous data sovereignty, right? And there was a lot of human biomedical thematic, but there was also biodiversity thematic. And it's another area where, when I say we want to develop technology and partnerships into that general direction, that's sort of an aspiration that we're trying to work into, but we don't have it yet. And I'm sorry that this continues to stall. I might just do like a wholesale refresh and try one more time in a minute. That's kind of all I have before maybe some questions. And thank you. Yes.
What was supposed to come out of that?
Oh, that last link?
Yeah.
Let me try one more time. Just a map that shows a relatively complex shapefile of the region that is assigned by someone, right? We got that shapefile from somewhere, and all of the plant diversity, like the records that are there. You know, so it's sort of the high level message is just, we have a huge amount of biodiversity information for any variously delimited part of much of the Americas at a minimum in our various databases, where the default is all of this is completely open, right? It was sort of built under the open data paradigm. There it goes, right? And I think we are now increasingly and also in a new phase of data ethics, right? And data economies and data governance. And that means, again, that our data infrastructures need to follow along. But of course, this is the technical issues are often not the ones that are the real bottlenecks, right? But in this case, the technical issues are also still somewhat of a new boundary that we're trying to push through. Yeah, and let me see if I can get the other record. It's a sunflower. Yeah, so we have protections here. You can see that the record is attributed to the Navajo Nation Herbarium, and they are publishing here on their account, right? But nevertheless, if you look at the image, we have things like a Navajo name, right there, and I don't know. And that's actually kind of an issue, right? Because I happen to be the person that could take that portal down in the next half hour if I wanted to, right? So it's sort of like, how did we even get here? And now what does this all mean? And that's just an area where we're curious and also active in finding new solutions.
So our lab maintains the biggest living collection of mycorrhizal fungi.
Mhm.
We don't manage our data very well at all.
Mhm.
So, and it's different because we're growing them out. We have to grow them out every year.
Yeah.
So the information on each of our accessions is dynamic in that it has longitude generations.
Mhm.
When we talk to, there's another museum database system.
Specify?
Yeah, so we talked to folks at Specify,
Yeah.
they couldn't accommodate
Yes.
us, so we
Right.
gave up on it.
Yeah.
Can you accommodate what we do?
We might be able to. Yeah. We're Specify and Symbiota are now both at KU. Which
You talk to each other?
it's more than that. Yeah. Yeah. Yes. Yeah. Yeah, it's one of the many fun parts of my job, right? We are legitimately thereby one of the top very few academic sort of leaders globally in this biodiversity service infrastructure design and provision field, right? And there's a lot of complementarity there that I feel like both softwares will continue to do what they do, and we can also find the synergies, and it's going to be wonderful and exciting. So one of the, if you have any sort of research collection, right, one of the first forks in the road that we usually ask about is, are you preserving this collection for enough of a time beyond your study that you need some sort of permanent record thereof, right? You might even discard samples, but do you need to preserve the data beyond just the initial time span of your research active program, right? And if the answer is no, then typically for Symbiota, that's not an ideal fit. But if the answer is yes, then yes. So from talking to the LTER communities, right, and posing to them the question, you take samples all the time, which of these samples sort of pass that threshold that you actually want to preserve them for an extended period of time, whatever that is, versus not, right? You can or maybe cannot imagine that that's already a very, very profound question to make decisions on for every single sample type that they have. But if that decision is, yes, we should, then we would basically try to see to what extent we can at least, your data will have some of these geographic, collecting event, taxonomic, and other components that fit with the Darwin core, right? And then there will be other things that are above and beyond what our scope is, right? And so what we did with the NEON main portal versus the Symbiota portal, we just identified basically the overlap. And that's where we built the connections and the integrations. and then other things need to live elsewhere, ideally, right? But what's the common thread there is this idea to basically consider a ultimately decentralized network of biorepositories that can talk to each other, vis-a-vis or versus trying to build one repository that perfectly fits all of the data that you have. I think the community in general is sort of like learning that we have too many very, very strong, very, very large projects that we can't just build one centralized item for. And so we need to sort of see that if 100% of your data have no great place to live, but maybe 50% of your data can live well there and another 35 can live there, and then the other 15 can't live anywhere. What works, right? Yeah. Try us. Yeah, I will send out this flyer. There will be clear information of how to get in touch with us. If you send us a help desk ticket today, I can guarantee you within 24 work hours, you will have a contact established. Right? We're good at that. Yes.
Thanks. And so lots of questions, but I guess central one is, you know, a lot of this is specimen based, but you had a little bit of this in the neon stuff. I'm thinking about observations. You mentioned iNaturalist, but also eDNA and things like that, where the tools aren't a thing, right? The tools are a bunch of sequences that came out of a soil or off of a plant, or research grade, for example, observations and iNaturalist. Can you talk a little bit about how that?
All of those are in scope, right? Okay. So we might say that Symbiota requires a kind of voucher, where the voucher can be a physical specimen, it could be an image, right? It could be other data, such as a DNA string, right? And so as long as you tie that form of a biological signal level voucher, right, with everything else, it should be in scope, right? And we also want to expand the scope to be more comprehensive in those kinds of areas, right? So again, I think the initial idea is let's consider and let's talk, and we're certainly willing to accommodate. One of our future sustainability goals is we now created a portal, and again, I'm not so sure whether this helps or not, but if you go to the main Symbiota website, there's a page about the portals that we maintain. One that we stood up not so long ago, and it was sort of a pretty big deal for us, is we have one that is now hosted, as you can see by this URL, on government premises, right? Most of our other virtual servers would be with ASU IT or KU IT. We don't do Amazon Web Services currently, but this one we were able to collaborate with the USDA to put on a .gov server with all of the restrictions that entails, and also with the 508 ADA compliance, accessibility, and other, a dual login. If you've ever logged into like a government website recently, you better have your cell phone and then some, right? So it does all of that. And so we are trying to get, for example, into germplasm collections that the USDA, we are trying to establish ourselves as one of the premier sort of biocollections data repository for the USDA. And in that vein, we will need to expand data capabilities. The USDA tends to be very mission driven, not necessarily standard driven, right? And so we're trying to grow into areas where we feel like it makes sense.
So in this, you said biocollections here, you mean living collections?
Yeah,
Because you have
potentially.
Fungal, so you
Yes.
have, so like for your fungal collection there, you must have then cycles, keep track of cycles. So you have done this before.
Potentially, yeah.
Potentially.
Well, it depends on what the USDA, I can almost guarantee you that we are representing a subset of everything that they have, right? In terms of on their local data systems, right? But they've also seen that there are trade-offs. And the USDA, like many of us, needs to sort of justify their impact, right? And this is a way for them to do that. They may still have quite a few data on local systems that we're not quite ready for yet, and we will need an additional contract, right, to maybe expand into that direction. Yeah.
So you had given the example of an individual student collection, but it was also housed in a formal biodiversity collection, right? So thinking about like quality control and,
Yeah.
you know, is there some piece there? So if you did have, you know, somebody that was providing collection information that, you know, may or may not be that
Yeah.
correct? It's
Yeah.
like,
Oh, sure. Yes. So the communities are self-governed, right? We don't own the data. We also don't own the software. We just provide services to it. It's open source. Anybody can do what we can do in principle. But one of the design features is because you have these communities that are not everybody, everywhere, all the taxa, all time, you know, all data types or so. Instead, you actually have communities that somehow do think of themselves like that, right? And we have found that that has overall a collective positive effect on data, you know, comprehensiveness and data quality, right? But the way to get into one of these portals is literally you say, for example, to our help desk, or there's a portal manager that's on the portal website. You tell them, I have this in this collection. Can you set up a collections profile for me to this day, right? And you will talk to a human being. Sometimes some of the portals have sort of boards, right? Most of them are not necessarily that socially sophisticated, right? And the Ecdysis portal in particular is just very much it's bringing in more people and more collections, right? And data that are not fit for use, but are exposed for use, right, can improve that way.
and you'd also mentioned the Humboldt extension, so thinking about collecting events. And so is there like kind of metadata behind that that describes collection protocol?
Yes, there can be. For the, you know, this is one of the sort of boosts or synergistic value that we've received working with Neon, right? The Neon portal is in many ways sort of a window into the future. What if a Symbiota portal were driven and designed by a very, very structured and maintained, you know, sort of metadata protocol community, right? And so then there are linkages to the Neon sample category protocols and so forth, and integrations. With all of this, we always have to pay attention, as everybody else does, of what kind of development and functionality do we want inside our software environment versus where are our boundaries, and we build an integration with someone on the outside because we can't do it all. That would be terrible software.
All right. Thank you very much, Nico.
Thank you.